Okay, so here we go, chapter 5, structure and function of large biological molecules. So just a quick little introduction here. You guys, hopefully, this sounds familiar. All living things are made up of the 4 classes of these macromolecules. You have carbohydrates, the fuel, lipids, right, which are in your cell membranes, and fats, right, energy storage, and also insulation. That's what I was looking for. Proteins, which do just an amazing amount of different things, and nucleic acids, which actually I think I would say are the least diverse. You either have RNA or you have DNA, okay, for your nucleic acids. So there's the 4. So within cells, small organic molecules are joined together to form larger molecules. And we have just thousands of covalently connected atoms in these molecules. And again, structure and function are inseparable. Why? Okay, these just some vocab words here that you should be familiar with. Polymer is just long molecules consisting of similar building blocks. So we have lots of repeating units. These repeating molecules are called monomers. Mono means 1. Okay, so the 3 of the 4 classes of molecules are polymers. Carbohydrates, proteins, nucleic acids. Which one are we missing? Lipids. We'll get to that. Okay, how are they built? It's called a condensation reaction, or more specifically, dehydration. So what must be coming off of the 2 monomers to make the bond? Water. See that? Dehydration. You'll get an H off one end of the monomer and an OH off the other monomer, and then it comes together to make a water molecule. Okay, to get more specific here are enzymes. And enzymes are types of which are the 4? Starts with a P. Proteins, okay? And they speed up these dehydration processes. So here's a protein that helps build these molecules. And it's used in lots of other reactions. So polymers are disassembled to monomers by hydrolysis. Anytime you see lysis or lys, that means a split or cut, so you know you're breaking down. So the reaction is essential for the reverse. So let's show you this. Oh, my clicks.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown, with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme.
Okay, and here's a nice just diagram to help you guys to make sure you know this. Okay, now let's break down each of the 4 groups. So what is the monomer of carbohydrates? Read it. Monosaccharides. Okay, what's an example? Glucose. Okay, so that's as simple as a carbohydrate can get. And there are a couple, there's a few monosaccharides. It's just not glucose. Fructose is one. Glucose and fructose come together to make sucrose, which then would be a disaccharide when 2 of them are stuck together. Then polysaccharides are the polymers of sugar, so we get long chains of the monosaccharides. So here's some examples of your polymers. You got starch and you got glycogen. So starch is pretty much for plants here, and this is great for storage as well as support. So when you guys are eating celery, the long stringy stuff is made out of, hold on, I haven't got to that. That's cellulose. So starch is great for storing sugar, for storing energy. So that's why potatoes have starch in them. What else has starch in them? What? Rice, yes. And we make our pastas from wheat. So it's just a long polymer of glucoses, so that's a good storage of energy. Now think from the plant side, don't just think from our side. So the plant also needs to store these energy sources. So like in potatoes, it's under the ground, and then the plant can use it when it first starts to grow again without having to do photosynthesis and get the sun's energy. Okay, so glycogen, hold on, is a polysaccharide in animals, and again, this also we store for energy supply, and it's mainly stored in our liver and our muscle cells. Here's cellulose. So cellulose is a major component of the tough wall of plant cells. So once again, that's celery, that stringy stuff you see in there. So it can be very supportive, and it's just very long. Okay, now there's a subtle thing here. There's an alpha and beta rings, and that's pretty common, not just in carbohydrates. And I'll show you, it's just they're built slightly different. Okay, chitin. This is also another polysaccharide, and it's in the skeletons of arthropods. And this guy is molting. So this is its old exoskeleton, and it's cracked open, and he's coming out. And this is what the larval stages do, also like for caterpillars, for butterflies. And that way they can grow, because their exoskeleton, just like with lobster shells, same thing, they're stuck until they molt it off. It also is in fungus, and we can use a lot of these things. Okay, so that's it for carbohydrates. Now we move to lipids. So lipids, there are basically two types that I really want you to be familiar with. And let's see here. They are nonpolar, right? So they are hydrophobic, right? They're not polar, they're nonpolar. So they'll hang out with each other, but they don't want to hang out with anything that's polar. And almost a lot of chemicals in our world have to do with water, so then therefore they're polar. So we're talking about three things: fats, phospholipids, and steroids. The first two I want to concentrate more on. So you should know that the fats are made up of three chains, you can see here, and a glycerol at the front end where it's gray. And they're still made with a dehydration reaction. So this is fats, and that's their shape there. And then we have a couple of different kinds. When the chain, the fatty acid chain is like this, and you see no double bonds, they're just all single bonds, that's called saturated, because it's holding as much hydrogen as it possibly can. Versus unsaturated, now it has a double or triple bond, and it's also going to make it kinky. So the shape is not going to be that nice, straight fatty acid tail because of the double bonds or triple bonds, right? It's going to pull those carbons closer, making it a little kinky. So you can see right here some examples. Now to also kind of memorize this for the everyday world is your saturated fats. They are easier to break down. This is kind of backwards. So they're easier to break down, which that sounds like that's a good thing. But that means you get all the energy out of them more efficiently. which means in a world like we have today, where most of us, not all of us in the United States, we definitely have still a lot of starvation and hunger problems in the U.S., but a lot of us have plenty of food, if not too much food. And so then what the doctors are suggesting to us is that we should eat this oil, the things that are liquid at room temperature. They're unsaturated and they're harder to digest. If they're harder to digest, we don't get the energy out of them, and they just go through our digestive tract and we get rid of a lot of it. Therefore, we're not making fat out of all the excess. So that's the deal here with it. So basically, we're just choosing a type of fat that gives us less energy, and because of our situation with having plenty of food, and trying to keep less fat off of us, because too much fat then causes all sorts of things like heart disease and problems with joints and diabetes and stuff like that. Now, if you were in a situation where you're not getting enough food, what is your better choice? Yeah, we want to go saturated. Okay, so that kind of just talked about a lot of what I was talking about. Down at the very bottom, though, you hear a lot about trans fats and stuff. So what we've also been doing is we are converting unsaturated fats to saturated fats by adding hydrogen. It changes the taste a little bit too. It's also easier to store when it's solid. So hydrogenating vegetable oils also creates unsaturated fats. Let me back up. A lot of our saturated fats come from animals, like butter, for example, lard. And then a lot of our vegetable oils are the unsaturated, olive oil, canola, all that sort of stuff. So then what they've been doing is that we have been making this reaction happen and turning unsaturated fats into, saturated fats into unsaturated fats with trans double bonds. These trans fats may contribute to more than saturated fats to cardiovascular disease. So you're seeing these on packaging, right, where it says no trans fats and things like that. So again, and just another concern. So the major function of fats is what? Energy storage. Do not get this confused with carbohydrates. Carbohydrates, of course, store some energy. Every bond does. But we break it down quicker and easier into fats if we're not going to use it. But there's some other things here. Cushioning and protection, insulation of organs is also the main contributor of fats. Now notice we're not talking about lipids in general. We're talking about fats that are one of the three lipids. You guys see the pattern here? Okay, our second one is phospholipids, and where do we find these things? In the cell membrane. You guys remember those the bilayer? Remember the fatty acid tails, which are non-polar, don't like water, so they go inside, remember? And then we actually have the glycerols is polar, so it's hydrophilic, right? It likes water, so they line up. So these are your little phospholipids. So we could get in trouble if we needed to lose some weight, and I'm talking in general, you guys. Not assuming, all of you look wonderful, so don't worry about that. But as we get older, we're trying to stay healthy. But anyway, the deal is when people are trying to lose weight and are on a lower fat diet, sometimes they're not taking in enough fat, and every one of their cells, their cell membrane needs it. So, and you can see from steroids too. And we do need a certain amount of fat layer also. Okay? So there's all those phospholipids. There's all those phospholipids, and you can see how they line up because of the hydrophobic tails and the hydrophilic heads. Okay, the third component is your steroids. So steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. So they have these ring structures. And I think last time I showed you testosterone and you saw that in estrogen. So cholesterol is another one. Okay, here's another thing that when we say, we might think, oh my gosh, that's so bad. No, you need cholesterol. Cholesterol is also in your cell membranes. Too much of it is when it starts building up in our arteries and causing problems. There's also there's a few cholesterols, there's a few different types, and some are really helpful to us, while the other ones can build up if we have too much. So it is essential, but too much can contribute to cardiovascular disease. Okay, now on to proteins. So proteins count for 50% of your dry mass. Remember how we talked about that last time? We're full of water, but that's not what you're built of. And then if we were to dry you all out, 50% of you are these proteins. There's the other things. There's the carbs, the lipids, whatever else I'm missing there. Now, huge variety here, huge variety of all the different things, all the shapes and all the different things they can do. So we have structural support, we have storage, we have transport, we have communications, we have movement, and we have defense. So here's a nice table here that talks to you more specifically of different examples of proteins. Okay, let's check this one out. Oh my.
Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
Okay, another type of protein are enzymes, right? And enzymes are catalysts, okay? When I rub my hands together, that heat is a potential catalyst. But to get reactions to happen in the body, we can't light the cells on fire. We can't get them to rub together. So chemistry has a magic thing, which are these enzymes. Okay, so enzymes make reactions happen at a faster rate, which makes the cell work efficiently. If we didn't have enzymes, the cell would die. It could not keep up with all the things it needs to break down and all the things it needs to build. So here's an example. Again, do you see how this is a beautiful example of form and function? They're very specific. One enzyme only controls one reaction. So they fit perfectly together with their substrate. So look at this, you guys. I want you to realize here. So what do we got here? We're going to start here with this product of sucrose, a disaccharide. Okay? And it fits perfectly in the enzyme. Okay? Then it just in how it fits, it puts pressure on the bond so it more easily breaks the bond. So now we have the products. Did I say that right? These are our products. That's our reactants, right? Notice there's a synonym here. In an enzyme reaction, we call our reactants what? Substrates. Do you see that? So if you're kind of confused with that vocabulary. We will have a whole little section on enzymes. We'll work with enzymes next chapter. Right now, I just want you to know that they help with reactions and that they're a protein. Okay, so polypeptides. We're moving on to proteins now. So these are polypeptides are polymers built of the same set of 20 amino acids. So the monomers of proteins are these 20 amino acids. Also, the proteins will be built of one or more chain, which that's the other word. A chain of amino acids is a polypeptide. We don't like, it'd be nice if we call it polyamino acids or something, but we call it polypeptides because the peptides are the name of the bonds that link the amino acids together. So here's your amino acid. They're exactly the same except for the R group. also known as the side chain. So people remember when you were back there folding your proteins, those 20 amino acids that you put on the little round piece of paper, those were all the different possible groups that are poking out here on an amino acid. So lots of variety. And that's why proteins are the most diverse, because they have 20 different building blocks. Okay, so here they are. Remember from that round chart, again, here's the non-polar amino acids, here's the polar amino acids. We have acids and bases, and that of course comes with some charges. And then they interact differently with each other. So that interaction allows them, those polypeptide chains, to be folded up into a fixed shape. They have a very specific shape by how they interact with those R groups. Okay, so there's 4 shapes of proteins, and lots of times they might be starting to be folded. So a lot of times when we say primary structure, this is just a loose chain of amino acids. I don't know of any examples of this. I only know of examples of it because they haven't been modified and folded yet. But I could be wrong. There could be a little bonus extra credit. Share it on our Google Classroom or our Facebook. Kind of like first person, to do it gets the points. Can't start till after 2:30 today. And also put your resource. And more than one person can get this bonus points, depending on, because they can find other examples. So I'll say up to 5 people can contribute to this. But you've got to not repeat anything, so that means you've got to read other people's. So what are you researching again? Is there such a thing as a protein that stays in primary structure, which is just a chain of amino acid? Okay? All right. Then we have secondary, tertiary, and quaternary. I'll show you some examples. So here's primary.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay, then secondary. Now when you guys are, oh wait, actually no, I got time. When you guys are doing your extra credit, make sure you don't do secondary structure of something. It looks like a long straight line because it's zigzagging pleated, or it's spiraling, and you could say, oh, maybe that's what she's talking about. That's just a line of amino acids. No, the primary structure is like a loose thing, a loose chain, a polypeptide. So let's look at secondary.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where 2 regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, so remember the helixes and the beta sheets or the pleated sheets, that's going to be secondary. So here's an example of pleated sheets with the silk of spiders. And then they also have spiral strands also, which make them like springs and elastic. Okay, third one, tertiary.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, and quaternary.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Okay, so quaternary, the big difference here is that it's involving more than one chain. So that means that that polypeptide chain could have been made by different genes, and we'll get to that later. So when you think back of our modeling of proteins, when you guys were using that, what do you call that floppy foldy thing we were working with, foam something, I don't remember. What structure were you making? So it was one polypeptide chain, that would be modeling one chain, right? It was continuous. So which one was it? Primary, secondary, tertiary, quaternary? Now there could have been a chance, depending on how you randomly made it, but were you in a complete helix made by hydrogen bonds, or still a secondary would be pleated, going up and down? I didn't happen to see any models like that. So what were you guys in then? Was it being held together in a certain shape? It should have been, with interactions between the R groups. So which one is it? Okay, I'm going to leave that one to you guys. Go find that one out. You should be able to figure that one out. Come see me if you need help. Okay, so they just showed us hemoglobin, and with one amino acid substitution it makes the hemoglobin dysfunctional. And it makes it disshaped, and it won't carry oxygen as well. But even more of a problem is it's sickle-shaped, right? So it makes the red blood cells sickle-shaped, and they don't move through our arteries, and that causes major problems. So here you could see the different amino acids that build this protein. We have one difference. This is normal. This is a mutation. It's no longer folding correctly because of the interactions, and then it gets messed up here. Okay, so a vocab word I want to make sure you emphasize. It might be new to you. Proteins are very fragile. If they get too hot, if they get around acids, if their light bothers them, those very loose bonds that hold them together in that shape will fall apart. If those interactions are not there, if those bonds, even though they're loose bonds, weak bonds, they'll fall apart. If it falls apart, can it do its job? No, it's not going to be misshaped, it's not going to fit. And that we call that what? Denatured. Okay, so that's when it loses its shape, so it's inactive. Here's a nice example. Now, we can have a bad situation. For some time, and because those interactions, most of them don't need an enzyme, right, to help them. So just like when you guys were folding it up, they're going to do it on its own. So sometimes they can be restored. Okay, there are some helper proteins that assist called chaperonins. Like think of chaperones. They're there to like assist and help. And here's a picture. And you guys, these are proteins. These are proteins too. And look how complex they are and how they're perfectly shaped to do their job. Okay, so how do we know what these things look like? We use X-ray crystallography, which means we take the proteins, we dry them out, and then they're kind of, we could say they're in a hard structure, and a lot of them look like crystals, so they called this. And then we can take X-ray images of them. Another method is with spectroscopy, I can't say it, where we shine light through it and see then how the light refracts, and we can get imaging that way. And then bioinformatics is pretty awesome because we're getting to sequence things. So we can sequence the whatever it is by what amino acids it's made of. If we know what amino acids, what the order is, then like you guys did with the modeling, we can have a hypothesis of how it would fold together because of these interactions, because the property of each of the amino acids. So those are some pictures of what I'm talking about. And again, remember these amino acids are put together from a gene. So there's this huge relationship, right, between proteins and nucleic acids. So the last one, we got two types, DNA and RNA. The difference, if you're curious right now, is this is missing an oxygen, so deoxyribo. This is a sugar. This is ribose, the sugar in RNA. Occurs in the ribosomes when we build our polypeptide chains. So the nucleic acids are the polymers of polynucleotides. We can also call the monomers nucleotides. And again, there's some redundancy here. Each one has a nitrogen base, the sugar, and the phosphate group. So here's our pictures. So notice, cytosine, thymine, and uracil, which is only in RNA, thymine's only in DNA. Look how similarly these two are shaped. So you can see how one can easily replace the other. Now, they can fit, A's fit to T's, so down here, so all the A's and T's will fit together, and C's and G's will fit together. So the pyrimidines are the cytosine, thymine, and uracil. They have a single six-membered ring. The purines are the adenine and guanine. They have a six-membered ring. Now, I memorized this backwards. Pyrimidine seems like a long word to me, yet it's the smaller ones. So I have it memorized backwards like that. Purines is a small word, but it's the bigger one. It has a six-membered ring with a 5. So go back, I'll show you this picture. So see what I mean? Pyrimidines are smaller than purines. We got a double ring here. And I said that. We're good there. Okay, so the linear sequence of the nucleotides, passes from parent to offspring. So your main function here is what? Heredity, right? Passing the code to offsprings. And we can use this to figure out who's more closely related to who, which also pushes all the way into evolution.